FLAVIO KAPCZINSKI. I am a professor of psychiatry in Brazil, and during this winter I'm here in Paris as an invited professor at Paris University, and the topics I am developing are related to the staging of severe mental disorders and the use of biomarkers in psychiatry. Bonjour à tous, je ne parlerai en français, mais j'apprends quelques mots. Peut-être l'autre année, je peux donner en français. Well. Marion, thank you very much for your introduction. Thank you for all of you to come here. Professor Tamuz, also it's a great honor to have you here listening to the talk and the friends I have made here in my brief stay. I will be talking about biomarkers, the use of biomarkers for staging in bipolar disorder. As Marion has mentioned, we don't have the idea that staging is useful just for bipolar. We are going to use bipolar as an example, and it's our area of expertise, but we think that many of those ideas are applicable to other severe psychiatric disorders such as schizophrenia, maybe autism, and surely addictions. I think the field of addictions is very interesting to apply this. Severe major depression, I think is good for the model as well. So we bring this idea just to remind you that in the brain we have always these two opposite poles. We have the possibility of losing neuronal connectivity and even losing neurons, but at the same time we have the possibility of protecting those neurons and those connections. And basically the brain tissue is not static just there. It is on a constant dynamics between losing connections or having new connections being formed. Next. But then, so if the neurons are changing all the time, what would be, the principle behind all this? And then the most important work has been done by the Professor Levi Montalcini. She died like 1 month ago. She was like 93 or something. And she was the first one to describe this family of factors. She described the NGF, the nerve growth factor. And nowadays we pay a lot of attention to the brain-derived neurotrophic factor, which is the most active neurotrophin in the adult. And much of the plastic changes that we see in the brain and maybe that are related to disorders such as bipolar disorder are mediated by those neurotrophins. Apart from the neurotrophins and apart from the possibility of losing connections and losing neurons, we also have adult neurogenesis that occur in certain places in the brain, the periventricular zonal area and the dentate gyrus of the hippocampus. So here you have one place where neurogenesis is occurring. We think, or we may think of the neural tissue or the neurons are cells that are receiving the pressures from the environment. Here you have a tree that is, you have the pressure of the wind and the tree is adapting. And the neurons as biological systems are not very different from that, so they can have new branches and they adapt to the stress that they face. In situations when you have a lot of changes, maybe neuroinflammations is one of those changes, you can have also a programmed cell death and then neurons are lost. there are various reasons for that, but one of simple one to explain is that some inflammatory markers, such as the TNF-alpha, binds to extracellular receptors in the neuron, and those receptors can trigger cell death directly. There is a death pathway. So depending on the degree of neuroinflammation you have this situation of loss of neurons. Well, let's come back a little bit to the area of bipolar disorder. When we had the descriptions by Kraepelin, which are the most important, of course, he wrote so many things. It's difficult to say exactly what was his thinking, because from book to book he was changing a little bit his ideas. But mainly, the legacy from Kraepelin is that bipolar disorder is a phasic disorder, so you have attacks of bipolar disorder, meaning depression or mania, and in between the episodes, people are working normally, and they can have a period of euthymia which is more or less free of symptoms. But then, in the last 15 years, we have seen that the literature has changed in our field, and we see that when people have bipolar disorder, they're detected with bipolar disorder, they already have some cognitive changes that make not all patients, but some of the bipolar patients different from controls. And then once those patients have like 12, 15, 20 episodes, then their cognition goes into a much poorer functioning. So we cannot sustain the idea of Kraepelin that bipolar is just a phasic disorder, but we should acknowledge that the bipolar patients that we see, they change with time. As the disorder progresses, they come from this cognitive function to this one, which is similar to the one that we see in some patients with schizophrenia. When during my talk, I always say some patients, some bipolar patients, because they, of course, are not a very homogeneous group. You have a lot of heterogeneity, but I'm talking about, kind of an average patient, an average ideal patient. Next. So, for instance, you have seen that previous slide on is a summary measure of cognition, but this is taken from the daily practice, right? This is one bipolar patient with 53 years of age. He had a few episodes. He was well controlled under lithium, and this is 59 patients, not much older. But you can see the shape of his brain. There's a lot of frontal atrophy, ventricle enlargement, and atrophy generally. And this is a very common thing to see among those patients who are in very late stages of bipolar disorder, patients that cannot take care of themselves. They need the help of their family, or they need a day hospital. When you do their brain scans, it is many times it's like that. And this is one of the reasons why people, when they say, well, there is some differences between imaging of bipolar patients and controls. Of course, if among those bipolar patients you have those late stage guys, the curves are biased towards them. And what we think is that not all bipolar patients, not those type of bipolar patients, but those type of patients would benefit from the input of neurocognition, neuroimaging, and the use of biological markers. Maybe not now, as we were speaking previously, but maybe in the years to come we will be able to use some of those inflammatory markers to tell us who are the patients who have a more advanced case of the disorder. Next. not only imaging or cognitive function changes in our bipolar patients as the disorder progresses, you can see that the response to lithium that one have until having 10 episodes is lost after the 10th episode. So some other treatments such as the valproate or atypicals may be still working, but lithium classically is a better treatment for more to initial stages patients who don't have so much comorbidities. So it is a special cluster of patients, and of course not all of our patients would benefit from lithium as we know very well. Next. Well, so this is about is a bit of a superficial talk. I cannot go in depth in all aspects, but I want to give you more or less the situation of our research, what we have done in terms of looking into biological markers in bipolar patients. Next. First, we had these findings that BDNF serum levels were lower in bipolar patients, and then we found that this was true when they were in acute episodes like in mania or depression, but when they were euthymic, the BDNF levels were similar to the level of controls. So I was talking about having this distinction between early stage and late stage patients, but when we look into biomarkers in bipolar patients, we have to pay a lot of attention in the acute stages, because many of the changes are happening there. And then, of course, when you want to use clinical or biomarkers clinically, you don't want to get patients in mania or depression. You want them to be euthymic and see what can you measure in this situation. So one of the possibilities of the deterioration in terms of the cognition and in terms of the integrity of the brain of our bipolar patients may be related to the BDNF story. So we know that stress reduces BDNF, mood episodes reduce BDNF, and this reduction could be leading into some degree of cell malfunctioning, and then a reorganization of the brain tissue. And then in this paper we talked about pathological rewiring of the brain. So, and this is what nowadays we tend to use the word Neuro Progression, which actually was introduced by this Australian researcher Michael Burke, and then we put forward a notion that maybe this neuroprogression is occurring through some of those pathways I'll be talking about. Next. So we started to work in some inflammatory markers. Next. And then we found that, for instance, TNF-alpha, there are some minor changes in the first episode, but when you have patients with multiple episodes, then there is a very, there's an important change in the levels of TNF-alpha. So in patients with multiple episodes, maybe the ones with that have progressed more, the disorder would have more inflammation. Next. The story is not similar to all biomarkers. For instance, the C-reactive protein, we didn't study in terms of staging, which is a good idea as well, but we did in the phases, and then we found that in mania you have a lot of changes in terms of the C-reactive protein. So again, it's this story of having acute changes in biomarkers according to the stages, not the stages, but the episode of the disorder. The same story we found using several markers of oxidative stress. This is to summarize, more or less, we found that euthymic bipolar patients had oxidative stress in levels which were similar to controls. And during depression or mania, you have an increase in oxidative stress. Not only oxidative stress, but oxidative damage as well. Damage to proteins, damage to lipids, and in this paper, damage to DNA, right? So you could see that those euthymic bipolar patients had a lot of DNA damage as well. This is not from our group, it's the group of Boston of Naomi Simon. And she showed also that there is telomere shortening in those bipolar, not only bipolar, she put together major depression, bipolar, you know, those are difficult patients, let's say, with mood disorders mainly. And those patients have telomere shortening, and the telomere shortening is a measure of age. So those patients had like 10 years, biologically 10 years more of age than their normal controls. So many times when we see our bipolar patients, particularly the ones that have are more late stage, we will see a patient who looks older than he is, right? And biologically, we can find that using those type of markers. Just coming back, as Frank Bellivier is working with addictions, in alcoholics this has been replicated recently. Next. Well, so summarizing, you know, this data, we found that neuroprotection is going down when you have patients with multiple episodes, inflammation is going up, and oxidative stress is going up as well. So if you think about the Ingmar Bergman dichotomy, life and death, you see that in those bipolar patients, late-stage bipolar patients, you are favoring more the loss of connectivity in the central nervous system due to less protection and more damage. Next. We did one analysis combining all those markers in different phases of the disorder, mania, depression, and euthymic. Next. And we put together markers of oxidative stress, like T-bars is damage into lipids, PCC is protein carbonylation, and then interleukins, BDNF, and combining, measuring the shared variance in those groups, we could see that the toxicity, let's say that deviations from the norm are toxic, right? And then we put, to make a case that it's toxic, we took a situation of extreme toxicities. Those were patients with septicemia, right? So those are those with septicemia. Those are the controls. Euthymic patients are similar to controls. And when they have mania or depression, they have this oxidative stress and inflammation going on. Maybe Tamusa would say that oxidative stress is a consequence of inflammation. And maybe that's correct. At that time, we were very enthusiastic about oxidative stress. Nowadays, we think maybe neuroinflammation is a more important hypothesis for us to look at to explain, you know, the deterioration related to bipolar disorder. Next. When you assess this toxicity, you can see this is the septicemia, right? And you can see that some manic patients have a lot of inflammation, like almost if they were with septicemia. Those are the ones with depression, and they are clearly different from the ones with euthymia, which are very close to the controls, but some of the controls, they don't have any inflammation, which is rare in the case of the euthymia. And then you see that even euthymic bipolar, they do have some degree of inflammatory changes going on. Next. Well, as we work in a basic science laboratory, we have these cell cultures, and then we wanted to see if putting the serum, if the serum of the bipolar patients is causing, is full of cytokines and inflammation. Maybe if you incubate neuronal cells with the serum, then you could change those neuronal cells. And those are very preliminary results, but which we are organizing to publish. But when you do that, you do have some interesting changes in those cell cultures. Basically, you have a pattern of endoplasmic reticulum stress, which is again an indication of cell malfunctioning. Next. Well, but when we talk about, this inflammation or this toxicity going on bipolar, it's a bit loose, how can we relate this in the literature? We know about stress and about major psychiatric disorders. The literature of stress has a good development and good grounds to make all this relationship. For instance, stress and increase of glucocorticoids can induce mitochondrial damage, which in turn can induce apoptosis, which in turn cause inflammation and toxicity. We thought of that because when you have apoptosis, even the silent cell death, it's not that silent. You recruit a lot of inflammation to the sites where there is apoptosis, right? So we thought of this idea to explain this toxicity going on in bipolar. Next. For instance, it's more or less consolidated in the literature, the idea that if you have an animal, this is a control animal, and this is an animal under chronic stress, and this is the frontal lobe neurons, then you don't lose neurons, but then you lose a lot of connectivity, right? And this is related to stress, but I think some of the lessons of the stress we can put into our models of bipolar and schizophrenia. Particularly bipolar patients, they tend to amplify the stress they receive from the environment. So we make a joke, we say that you get to these bipolar disorder patients more deteriorated, and then you say hello, and they say why? They don't say hello as well. They are always worried that something is threatening them, and of course, if you expand this to the daily situations, everything is a bit more stressful for them that it would be for a normal person. Next. And then in the stress field, not in the classical psychiatry field, but the stress researchers, they like a lot this concept of allostatic load. More or less, it says that all adaptation comes at a price, right? If we eat a lot of fatty foods, we can deal with this perturbation of our equilibrium, but then we have to recruit a lot of homeostatic mechanisms that, if they are overused, they start to produce some sort of imbalance. Let's say like marathon runners, they adapt to the run, but then if they run too much, they start to have joint problems and things like that. And this idea of allostatic load we applied into major psychiatric disorders, bipolar particularly. We thought that if you have the stressors, which are the arrows, plus the episodes, right? And each episode is a stressful situation. You can have this load of allostasis going on, and then you have like a scar of allostatic scar that would be the interval dysthymia and the cognitive dysfunction that you find in bipolar. So this is one working hypothesis. Is interesting, is nice, but it's somewhat vague. I like this because we don't have anything better, but I would like to be in the position of saying that in bipolar maybe we have a multifactorial story, but we have more weight of one factor, such as inflammation or this and other. We need to be more specific, otherwise we cannot tailor any treatment to help those people. Well, not only bipolar patients have these cognitive problems, but they have more mortality than the non-bipolar, particularly if they are not treated. The non-treated have a much higher mortality than the treated ones, particularly of suicide, of course, but then they have higher mortality in terms of vascular disorders as well. So treating the bipolar patients seems to protect them from early mortality. And the North American statistics say that type 1 bipolar patients, they tend to live like 20 years less than non-bipolar. So it's a very important reduction in the life expectancy. Next. So if we are talking about the brain reorganizing, or this type of brain rewiring that we say that we call neuroprogression, we could have this story of stress, glucocorticoids, but also maybe neuroinflammation, and then some mechanisms such as mitochondrial dysfunction, synaptic apoptosis, and then the reorganization of the brain. Why we talk about brain rewiring and neuroprogression? Because we cannot talk in bipolar about neurodegeneration. It's not like having Alzheimer's. So it's a different story, but it changes the shape of the brain. So we think we need a new concept. That's why we are putting forward this. We thought that for this neuroprogression we would have certain pathways that would be important for us to study. The inflammation, of course, the excess of neurotransmission of glutamate and dopamine that can be toxic, as you all know, and the BDNF, or the neuroprotection story, which is the opposite to the ones that are producing the damage. Yeah, here you would have interleukin-6, TNF-alpha, and maybe other players. And one common pathway after inflammation and after these intracellular changes is the increase of oxidative stress, which we think that may be playing an important role. Next. Related to oxidative stress, it's important to remember that when you have intracellular changes, or even if you have things such as neuroinflammation, the mitochondria are the organelle that is perhaps more sensitive to stress or to changes in the environment, because it's the center of the energy. So if you have any malfunctioning within the cell, the mitochondria will open its pores to initiate a cascade of apoptosis. So a cell, maybe the cell thinks that, well, it's better that I will die alone than to cause necrosis and kill everybody, right? So it's like a defense. When you have these changes, the mitochondria is activated and unleashes a cascade of apoptosis. Next. When you have other types of tissue damage and you open the pores of the mitochondria, you release in the circulation dumps, which are products which should be inside the mitochondria. The mitochondria, as you know, are thought to be a bacteria, perhaps, that got into the cell and then developed as the mitochondria. And much of the content of the mitochondria is very, it causes a lot of inflammation when it is outside. And I'm putting this slide here because the latest thing that we measured in our bipolar patients when they are manic or depressive is these dumps, which include mitochondrial genetic material and other stuff that causes a lot of inflammation. And much of those factors are increased not in depression but in acute mania. Next. Well, some of you will think, you are showing these changes in blood and inflammation and all these, but this is related to the peripheral blood. It's not related to the brain. And then I got this interesting study to show you. When you make an homeostasis of a young rat with an old rat, this is the brain of the young rat before the homeostasis. When the blood of the old rat circulates into the brain of the young rat, it loses a bit of its neurogenesis. This is a lot of neurogenesis. This is losing a little bit. So what circulates in the peripheral blood seems to affect what is in there. Things such as neurogenesis and surely the well-being of the neuronal cells. The opposite is also true. When you take the neurogenesis of the old rat and you make the homeostasis with the young one, you increase neurogenesis. So maybe in the future, if we think about reducing this excessive inflammation that is going on in our bipolar patients and we do it systemically, we are also helping the brain cells of those patients as well. Next. Well, so this story of neuroprogression, or why patients have a progression in their disorder, right? Maybe is going to be important in our models of causation in terms of psychiatric disorders. This is, people like a lot the ideas of Avshalom Caspi. He's saying that, you know, it's about gene and environment. Gene and environment interact, change the working of the brain, which then in turn causes the disorder. Next. What we are saying is that part of the problem is that disorder is causing changes in brain, right? So it's like a vicious cycle. Once you have this disorder going on, you have, next, more and more of this one. So this is early stage. Then in early stage you have higher load of the genetics going on. And in late stage cases you have the genetics, but also you have the effect of the disorder itself, each episode causing more and more changes in brain. So I think this is the important part of the story. For us who treat severe mental disorders such as schizophrenia and bipolar, we do know that it's important to get patients out of the active phase of the disorder, the schizophrenic patients to stop to be so psychotic and stopping to listen voices, and for the bipolar patients to stop to have the acute episodes. Next. If this makes sense, if we believe that once you have a major psychiatric disorder, it can be more and more severe, it can cause more and more deterioration, then you cannot think of bipolar patients as being all equal, not even the schizophrenic patients as being all equal. you have to have in mind that you have an initial or maybe a latent at-risk phase, an initial phase, and a more complicated late phase of the disorder. And then we come to the idea of the staging. Why staging? Because we cannot accept anymore that we will have one algorithm to treat all bipolar patients, and we would not take into account the idea of the progression of the disorder, because then it's good to give lithium to everybody, and we do know that if you take a late stage patient, this is not a good idea. So we think we need staging in psychiatry. What type of staging? We were discussing this before. We are not very sure, right? I have put forward a model for bipolar. Our colleagues from Australia have also a model, and Giovanni Fava has a model, and so there are many models. We think that using groups like this one of experts, we can have the data set, and then we show, the model. We don't create from our minds. It's driven by data. Nowadays, we don't have this data yet. Maybe you guys in 5 years' time will be able to tell what is staging in bipolar. But for now, what we have found is that it's clinically useful to distinguish people who are at risk. Here is not risk, it's ultra high risk. It's people who have first-degree relatives with bipolar, and they already present symptoms. They have, generalized anxiety. They may have attention deficit disorder, or some type of suffering that has not reached the threshold for bipolar. Once we reach the threshold, we say, well, this is bipolar type 1 or type 2, right? Because we have either mania or well-defined hypomania. What is a stage 1 patient in our system? Is the Kraepelinian patient who is ill in the mania and depression, but it's well in euthymia. So this is the patient from is the ones that we like to say, well, I have a patient that is very successful, he's doing so well, and so on. Maybe they are not the majority, but many of our patients with good treatment, they can work properly, they can be quite successful, and they can have a good family life, and basically they are living normal lives. These for us are stage 1. Stage 2, those are the patients who have reversible problems, things such as they are not normal during the euthymia because they are drinking too much, or they are having panic attacks, or they have some personality disorders that doing some psychotherapy or some treatment of the comorbidities, we can get them back to stage 1. Stage 3 are the patients who cannot work anymore because of the damage induced by bipolar disorder. For those patients, we may have to write to the pension system to allow them to have an early retirement. And many of them, they want very much to work, and they cannot work, as they were educated. For instance, if one has been educated to be an engineer, he cannot work doing calculus, complicated things, but he can work in a bureaucratic position, right? But you have a marked decline in the working ability. By stage 4, we mean those patients who cannot take care of their own finances. Those patients are a little bit like, early Alzheimer's, perhaps. They have to rely on their family, or they have to have a day hospital. Typically, their family members would say, well, he's more or less okay, but in his room nobody can get in because it's such a mess that it's just impossible. Or they will say the only problem is that when he makes his shopping, he pays a lot of money. Sometimes people take advantage of that and is always creating problems. So you can see that they are not able to live an autonomous life any longer. When I talked during this talk about biomarkers and about the findings in imaging, they tend to be more confined to stage 3 and 4. Particularly this stage 4 is very strong in terms of those biomarkers. Of course, I've shown also the biomarkers that fluctuate according to the phases of the illness. And that is why, to use this staging system, you have to have euthymic patients, or the closest you can get to euthymia. You take like the last 2 years of the patient and you think what is the closest, because some patients, they don't reach euthymia anymore, they are ill all the way through. So you have to try and find what is their best euthymia-like state, and then you do the staging. So I think we have discussed a little bit about the idea that the brain reorganizes during the course of severe mental illnesses. We have seen that some biological changes that we can measure in blood, like oxidative stress, inflammation, and changes in neurotrophins, may be part of the story. And finally, we got to the point that if we accept that severe mental disorders have a progressive course in many cases, then we have to accept that a clinical staging system would be useful for us. In psychiatry, whenever we talk about staging and doing more, ordinary medical things, people always are scared. Well, this is going to complicate even more, it is difficult. But if you think about this staging system, it's very similar to the one that the rheumatologists use for arthritis. They have a staging system, a pathological staging system of arthritis and a functioning staging system of arthritis. For them, more or less, I'm simplifying, but stage 1 would be like pain. Stage 2, pain with a little bit of impairment. Stage 3 cannot move properly, can have to lose the work sometimes. And stage 4 is like a wheelchair, right? So I don't think it is terribly different from our scenario, and maybe if we put together the data that we have, the data that you are producing in this network, in a few years we will have a consistent staging system that will help us to help our patients. Okay.